disease of dairy cattle, which is the most considered as the most important disease in the world as far as dairy cattle is concerned.
This call is
And
now being recorded.
subclinical
This call is
mastitis
now
is the
being
disease,
transcribed.
a
This
category of
call is now
mastitis which is
taking
not
notes.
grossly visible with no clinical features, but it's the most economically important because since the clinical signs are absent and it's often being left undiagnosed, leading to very heavy economical this losses to the farmers. That's why subclinical mastitis, since the clinical signs are absent, so it needs reliable diagnostic techniques for its detection. Now, commonly we detect subclinical mastitis by common cow side tests, like it can be California mastitis test, somatic cell count, and electrical conductivity. But in this research, we have tried some new inflammatory markers which are released in the udder itself in case of subclinical mastitis, and the detection of those inflammatory markers, it can pay another this way for the diagnosis of subclinical mastitis in dairy cattle. So these inflammatory markers are the acute phase proteins. Normally, the acute phase proteins are secreted by the liver under inflammatory conditions and they are present in the blood. But in case of subclinical mastitis, these inflammatory markers which are present in the blood, they cannot be taken as the reliable markers, because only those acute phase proteins which are locally secreted by the inflammatory tissues, like in case of subclinical mastitis, certain acute phase proteins or inflammatory markers, they are produced in the udder tissue itself upon inflammation. So if these particular diagnostic markers, they are detected in the milk itself, they are considered as much more reliable and specific diagnostic markers of subclinical mastitis as compared to the acute phase proteins if they are detected in the serum. That's why in this study we have tried to detect the acute phase proteins in the milk rather than in the serum. And the literature as far as India is concerned, it's very scanty, because whatever the papers I Googled, I could only find the acute phase protein studies. They have been carried out only in case of blood, but absolutely there has been no one paper I could found in which acute phase proteins they have been detected in milk in case of subclinical mastitis. And since there are many acute phase proteins, we have only chosen haptoglobin and amyloid A because these are considered as the major acute phase proteins in case of cattle. And as far as the diagnostic, to confirm the diagnostic criteria, they have to be compared with some gold standard, and the gold standard, it has been taken as bacteriological culture, because this bacteriological culture it's considered as the gold standard for mastitis, especially the subclinical mastitis, since the clinical signs are absent. Now we took 410 this quarter milk samples, and this is the data from where we collected the samples. And out of 410, 174 samples, they were positive on bacteriological culture, and they were considered to be the cases which were suffering from clinical mastitis, sorry, from subclinical mastitis. So 174 samples were detected as on bacteriological culture as positive samples for subclinical mastitis. Now, these milk acute phase proteins, they were assessed by the commercial ELISA kits. Two kits were procured, one for haptoglobulin and another for milk amyloid A. The procedure was carried out as per the kits protocol, and this cut off the lowest detection limit. It was calculated as per the kits protocol and the formulas which are applied to the standard ELISA procedures, sir. These are the results of cultural examinations we carried out, the cultural on various different bacteriological media like this mannitol salt agar, EMB agar, and nutrient agar, and blood agar, and then detected the samples which were positive for subclinical mastitis. And finally, the statistical this evaluation was carried out, and receiver operating characteristics were formulated for this acute phase proteins, which were then compared with the bacteriological culture for the finalization of the validity criteria of the results. So this is the diagnostic accuracy of the milk phase proteins when they were compared with the bacteriological culture. The sensitivity, specificity, positive predictive value, negative predictive value, accuracy for milk amyloid A and milk haptoglobin, and they are shown in the table with Cohen's kappa, which is another important this diagnostic this agreement of the diagnostic criteria or the validity for the tests, and it came out to be 0.82, which is considered as highly reliable and in agreement with the gold standard test. And for the HP, it was 0.61, which is considered as substantially accurate. And the ROC analysis, they revealed when the area under curve it was formulated for milk amyloid A, it came out to be 92.8% this area under the curve, and for haptoglobin, it is the 81.4% with a predictive value of 0.001. And all these values they were derived by the Mann-Whitney U test. This is the boxer plot of amyloid A.
Conclude,
Sir,
please. Yeah,
yes,
conclude.
sir.
Yeah, time limit is there. Conclude, please.
Yes, sir. And this is the box plot for the haptoglobin. That was the box plot for MA. And this is the ROC curve for this both of these acute phase proteins when they were compared, sir, with the bacteriological culture. And this ROC curve, it clearly shows that these are way ahead of the common diagnostic techniques which are used for the detection of subclinical mastitis. So we can conclude that acute phase protein estimation in the milk, they have a significant diagnostic value which deserve attention for SCM detection. So they should always be used in association with either we can go for the California mastitis test, we can go for the bacteriological culture, and then but these two tests, they have a superiority over those conventional tests which we carry out. And the recommendations are that since the kits are costly, so we have to prioritize the development of indigenous and user-friendly diagnostic tests which are based on milk acute phase proteins, especially amyloid A, because it has superiority over this milk acute phase protein. Thank you very much.
Any question?